Alright, so this week we're going to be wrapping up the idea behind how to market proteins and how they're basically used in medicine. So the goals for this week's lecture are how to harvest proteins and how they're used in medicine. So by the end of this lecture, you should be able to describe how to harvest proteins, how to purify proteins, and relate that to quality control. So in the context of quality control and testing, and be certain that your product is a pure product. And secondly, how you should be able to describe how proteins and biotech products or pharmaceuticals can be used in terms of the context of health. So we're mostly looking at vaccines, but we'll have a couple other examples as well. So those are the two goals for today's lecture. So right off the bat, how do you harvest protein? There's basically two methods that we're looking at. The first is filtration. So this is the idea that you're mostly doing by size. You're looking at concentration gradients. So the idea of high to low, go with the flow. So you'll try to get a protein by itself by using something like dialysis tubing, and we'll kind of have an example of that in class. The other is chromatography, and chromatography comes in different forms, but the idea is that you can separate based on different features. And we'll talk about some of those features on the next slide, but there's different versions ranging from column chromatography, which you might have done in chemistry, where you separate things using an elution buffer, or chromatography, which we're looking for a color change, which may be something like a plate where we're looking for a color change that shows up. This could be also things like electrophoresis, which build on this idea of chromatography, where you're creating an image graph that's using with colors. So there are different types of chromatography. Like I said, you can separate based on size. So whether it's something large or small, similar to how electrophoresis separates by size. You can also look at charge, positive or negative. This is especially true with things like proteins, where you can have different ions based on the amino acid content and character. Lastly, the last two would be affinity. So you can create pockets similar to an enzyme where products of interest will bind. They might also bind because of antibodies. So if you need a refresh on antibodies, that's in the last lecture. But the idea is that you can grab stuff based on its shape. And lastly, you can grab things or separate things based on hydrophobic characteristics. So this would be polar and non-polar substances. If you remember from chemistry, the idea like dissolves like. You can separate things based on what they tend to bind to. In terms of quality control, the idea is that you want to create pure products, and you want to be able to you want to be able to test its effectiveness. And one of the big things that you have to think about when testing effectiveness is that you want to have double-blind studies. So this means that both the researcher and the subject don't know. And this is really important because researchers can sometimes shade the results. And the way they can do that is through random generation, so using computer programs that don't tell them who gives what, using only the coding that they can then go back after and then find out who did what. Also related to quality control, we're looking at purification and effectiveness, which is also regulated by the government. Which brings us to kind of the last topic, which would be patents. So patents can really impact development of something. If something's open source, lots of people can use it, versus high patent stuff. And one of the huge examples outside of technology for this would be Disney. Disney is a high patent thing. This would also be kind of the debate between Apple and Android, whether you're an open source or a high patent kind of idea. Same thing with Wikipedia. There's a lot to be debated on this, and it's something that if we have time we'll hit on this in class. So to summarize product harvest, the main ideas that we want to make sure you understand are that there are multiple ways to separate proteins. Once you've kind of smushed it up and harvested, so this would be you're breaking cell membranes or crushing stuff up. There's multiple ways to separate the proteins, and also that this process is pretty highly regulated. You really want to get high yields, and you want to have a product that you can then give to somebody else. So that kind of wraps up the idea of product harvest in kind of a nutshell. Don't forget to reference your textbook and your lab manual for more examples and details. So next on, we're going to move on how to make pharmaceuticals. So last week we talked about how a lot of these things sourced are from the rainforest, but not everything does. And sometimes you'll find something in the rainforest, and you'll actually want to make it similar to the existing. So these are two ways to make pharmaceuticals. So you can find something, and you could actually match a receptor better. You can make it last longer, or remain in your bloodstream longer. This is the case for a lot of medications where normally your body just kicks it out, but you can modify that and make it last longer or have a higher intensity. So that would be kind of taking the idea of taking something that exists and making it better. The other thing you could do is you could make something completely from scratch. And so that would be through peptide or DNA synthesis. And this uses special machines to physically build it one by one. So this would be where you knew the sequence. For example, maybe it was like alanine, glycine, proline. You could make it and build it that way, which could then be made in a bacteria or another form. So once you've made kind of your pharmaceuticals and these things that you want, one of the key examples that people always bring up is vaccine development. So in vaccine development is pretty key because we're looking to create cures and make a difference. And so there's a lot of money in vaccine development, especially when stuff comes up that's pretty dangerous to the population. So the first thing you have to do is ID an antigen. And an antigen is a protein of the bad guy. And so in this antigen, you want to kind of look for something that's harmless, doesn't cause problems, and is pretty common. So this is the explanation for why the flu vaccine changes every year, because every year there's a different mutation and a different antigen that's a better match. And so what does the vaccine development create? It creates something that's injectable that stimulates the nervous system. The nervous and immune system. So it stimulates the immune system, specifically your B and T cells. And so this creates your own natural antibodies. Which allow you to fight off things for yourself. And this is kind of one of the treatments that's getting really big in cancer, the idea of using your existing immune system to fight off your own cells. So a summary of the use in medicine, the idea is that we want to be able to create peptides that can improve quality of life, and that can either be de novo, brand new synthesis, or a modification of existing things. Modification. We also learned in the summary that one of the big key examples, so the case study, the one you want to remember back on, are vaccines. And the reason why we're picking up vaccines is because there's something that's relevant to biotech. It's something that makes money. It's relevant. And it's something that's in current events. Every year, whether it's swine flu, whether it's Ebola, there's a disease that's going on that would help if we had a vaccine or treatment or a way to ID it or an assay for it. So that's why that's really key. So lastly, here's some review questions to kind of wrap up the lecture. You might want to think about these things. So what are the different ways of purified protein we hit on this beginning? What are factors to consider when testing a product? Describe how pharmaceutical products can be found, improved, or synthesized. And lastly, how can biotech products improve your quality of life? So try to process your information now that the lecture's done. Create a flowchart, a diagram, graphic organizer. It can even be a comic. It could be a webcast. You want to make sure that you're taking the time to explain and think through what you just learned. Make sure you get some good night's sleep as well. So lastly, check your book and your lab, use them as reference. Try to make connections and link everything. So biotech is really related to everything, whether it's your other science classes, whether it's a class you took in the past, whether it's current events, whether it's your future. Try to link everything to kind of make some strong connections about this. So hopefully you learned something from this lecture. If not, shoot me an email, try watching it again, try to find a way to process it. Of course, you're always free to reference your textbook and your notes. Feel free to give me a call or shoot me an email if there's anything that you're lost about. Otherwise, take care and hope you have a good one.